Yeah, I will
All right.
share the screen.
All right, thanks. So I want to welcome everybody to this week's Cancer Center Symposium. It's my pleasure to introduce Dr. Fuyang Xing today, today's speaker. So Dr. Xing received his PhD in electrical and computer engineering from the University of Florida in 2017. And that was the same year he was recruited to our campus, where he's currently an assistant professor in the Department of Biostatistics and Informatics at the Colorado School of Public Health. Dr. Xing has a very prolific publication record in the area of biomedical image computing. He's published in leading journals such as IEEE Transaction Medical Imaging, JAMA Network Open, as well as leading computer vision and medical imaging conferences where they have competitive conference proceedings. So in some of those conferences, they accept anywhere from as few as 5% of papers to 25%. So it's very competitive, and Dr. Xing's been very prolific there. The other thing I just want to give an advertisement for is he's also obviously a professor at our campus. He'll actually be teaching a class on Python programming in the spring. So if any of you guys are interested, you can follow up with him about details about that. And just as a format, I'll be monitoring the chat box today. So if you have questions for Dr. Xing, so please put those in the chat box. And then, at the right time, I can stop him and sort of ask those questions as the moderator. So with that, I'll turn it over to Dr. Xing, who'll be talking today about medical image computing with deep neural networks. Dr. Xing.
Okay, thanks. Thank you, Debashis, for the introduction. And hi, everyone. It's my pleasure to share our research, which is focused on the deep learning-based medical image computing today. And this is the today's agenda for the talk. And first, I will briefly introduce the background of medical image computing, and especially I will briefly introduce the application of deep learning in the medical image computing domain, and briefly introduce how the deep learning works as well. And then we will discuss in more detail how to develop the deep learning algorithm for different kinds of tasks, such as the cell detection and Ki-67 labeling index assessment, as well as the lesion detection in the paired image data. Okay. So probably you probably have known that for nowadays we collect many images in different types of formats: CT, MRI, ultrasound, and microscopy in the hospital or in the research lab. So for example, in one muscle research lab, we can collect more than 1,000 microscopic images per day. So with this kind of image data, the radiologist or the pathologist, they will inspect the images, measure the morphology changes in the image data, and or calculate some kind of biomarker to provide support for the disease diagnosis. For example, this is the one example image from the breast cancer from the breast tissue. And from these images, we probably can see that there are a lot of the tumor nuclei in these images. And the pathologist probably will get a conclusion saying that this patient has some kind of breast cancer disease. And probably for the humans or for pathologists, it's kind of more straightforward to saying that this patient has some kind of disease. But for the computers, it's not that straightforward. They only see the numbers. In the computers, each pixel will be represented by a set of values, such as for the RGB images, they will represent by 3 channels, and with 3 values for each pixel. And how can we extract useful information from this kind of numbers, such that we can provide support for the diagnosis? And this is the job for biomedical image computing. And biomedical image computing is a field to develop computational methods to automatically analyze and interpret the image data. And the goal is to provide support for the new biological knowledge discovery, clinical diagnosis, prognosis, or the decision support. So actually, in the medical image computing domain, you can find a lot of applications or tasks, such as the automatic cell detection or the per-state segmentation, and also some other image registration. And more recently, probably you will find some literature reported that how to generate a medical report or the test description giving an image. So doing this in an automatic way. So actually for the deep learning, it has already been involved all these kind of tasks. And in many applications, deep learning have shown better performance than the traditional machine learning method, such as SVM or random forest. So probably you want to have a question that's why deep learning is so popular nowadays. One potential reason is that compared with the traditional machine learning method, deep learning can consistently improve the performance when you have more and more data, because as long as you have a large enough and a sufficiently complex model, it can be handled this kind of complex data. But traditional machine learning, it will get very easy to approach their model capacity when you have a more data set. So this is probably one of the potential reasons for the deep learning is so popular nowadays. And before we go to the details, how to develop the deep learning algorithm for medical image analysis, here I would like to briefly introduce how deep learning works use a simple example. Okay, so this is one simple example. Here I show a very relatively shallow model. It only have 2 hidden layers. And this is a fully connected neural networks. So for in each layer, each neuron will connect all the neurons in the previous layer. And in each neuron, it will first do the linear combination of activations from the previous layer, and then use a nonlinear activation function to process the information. The reason why they need to use a nonlinear function, because if you can get many linear functions, it will be equal to a single linear function. You can do the method proof for this. So using the nonlinear activation function, we can process more complex data and model this kind of complex patterns in the data. Okay, this is the kind of very basic pipeline for the deep learning model. Before we can apply this model to the different tasks, we usually need to do the model training. Suppose that we are doing the supervised learning, and the typical pipeline for the model training is that first you get input and then calculate the activate function for each layer, which is what we call the forward propagation, until you get the last layer, and then you calculate the loss. And because, for example, for these images, you want to predict the image is a cat or non-cat, and then you can consider a sigmoid function as the last layer to predict a value between 0 and 1, and indicate that how likely this image is a cat image. And then you will measure this loss between the prediction and the gold standard, and back propagate this loss to the previous layer until the first layer, then do the parameter update. So this is one iteration for the model training, and then you will repeat this procedure many times until the training converge. And after the model training, you will be able to deploy your model to the user, and then for the testing or inferences. And in the testing stage, you only do a single pass for propagation. And hopefully your model can predict that this image is a cat image based on the model you learn. So this is kind of the very basic idea for the deep neural networks, and you need to train the model and then use the training model for the testing. And this is kind of the traditional deep neural networks. And when we talk about the image analysis or computer vision, when we say the deep learning, usually will mean convolutional neural networks. And compared with the previous one, the CNN model will have a convolutional layer and pooling layers. And each neuron in the convolutional layer will connect only a local region in the previous layer. It does not connect all the neurons in the previous layer. In this case, you can significantly reduce the number of parameters. And otherwise, you will need much more data for the model fitting. Otherwise, you will get it's very easy to get for fitting. And additionally, it also have a covered pooling layer. Although currently many people don't like this pooling layer, they use some other layer, such as the structure convolution, to replace the pooling layer. But in the beginning, this kind of standard CNN model usually have this pooling layer. The pooling layer will reduce the size of the feature map. And in this case, you can speed up your model training because you reduce the space, the searching space, right? And also reduce the requirement of the memory as well. In the last few layers, usually you will be use the fully connected layer for this kind of classification task. And this is the same as the previous slides. And in the last layer, you just have a number of the neuron, and then the number of the neuron will be the same as the number of the classes you have. And each neuron will predict the likelihood that how likely this image is a cat or a truck or a cat, this kind of the architecture. So this is a typical CNN model. And in image domain, CNN model or its variances of fully convolutional network are the most popular architecture, although some people consider some other architecture, but this kind of dominate technology for the CNN. And here I would like to introduce a few very powerful and state-of-the-art architecture for CNN, because in many medical image analysis applications, you probably don't need to build your own CNN architecture from scratch, but you just need to download this kind of pre-trained model, which is trained on the ImageNet. That has more than 1 million images for model training. So this kind of model is well trained. You can download this model and fine-tune this model on your own data set. In this case, you will not require a large amount of data for model training, right? So this is kind of the, and of course, you will need to revise maybe the last few layers because maybe the last layer is that you don't have the 1,000 classes based on your data set or the task. So you just need to revise the last few layers, and then you can do the fine-tuning. And this, especially for the VGGNet and GoogleNet as well as the ResNet, these kind of architectures are still popular in the medical image computing domain today. And a lot of people use this kind of architecture for fine-tuning. For example, this is one architecture that, one example, use the GoogleNet for the skin cancer classification. They want to classify these different types of skin cancers. This is a very straightforward application of a deep learning or CNN model. Here it uses kind of the IMSETN3, which is a variant of GoogleNet. So in here, in the last layer, it just replaced the 1,000 layers, use some custom layers, probably less than 1,000 neurons to predict the likelihood of this image, input image. So what's which type of the cancer of this skin cancer? And another application you also gonna find that is also use GoogleNet to develop automatic algorithm for the diabetes retinopathy in the retinal fundus images for this one as well. And this one also have the very straightforward application based on GoogleNet. And more recently, for the COVID-19 screening, and probably the current diagnosis of COVID-19 is not based on images, but in the initial presentation of COVID-19, the imaging plays a very important role in this kind of confirming the positive COVID-19 patients. And this paper or this study built this architecture to differentiate the COVID-19 disease from other lung disease, such as the interstitial lung disease. Here, they do this kind of queer screening based on CT. And this architecture is based on the ResNet or the residue learning block. Of course, here it used some kind of more additional complex module, such as the prior attention module, to further improve the architecture. And you can find the more details in this paper. And we will not go to all the mathematical details in here today. In addition to this kind of applications, you can also find many other deep learning applications in medical image computing, such as the survival analysis based on images. Probably will combine with the genomics data as well, and also do the image supervision as well as the content-based image retrieval. You can find many discovered applications of deep learning in the medical image computing. And I will provide a few discovered survey paper here. You can find many of them in the paper. Okay, so this is just some quick introduction for the medical image computing. And next, I would like to discuss in more detail how to develop deep learning algorithm to different kind of tasks in our research. The first project I would like to discuss is how to develop algorithm for the automatic cell detection in cross-modality microscopic image data. In this pathology, usually we can have a different type of the microscopic image data, such as discover the H&E stand colon cancer or Ki-67 immunohistochemistry stand pancreatic neuroendocrine tumor image data. So this data look very different. We want to develop an algorithm to automatically to locate all the nuclei. You can image that for this kind of image is very time consuming to this manually. So we want to develop algorithm to do this task. And probably you want to ask this, so why do we need to do automatic cell detection? This is one example or one application. Suppose that we want to classify all the cells. We want to classify whether the cells are tumor or non-tumor, because this kind of the information can provide us information for the disease diagnosis. So in this case, you will need to do the cell detection. In addition to this kind of the cell classification, cell detection can also support for many other studies, such as the cell tracking, also segmentation, as well as kind of the other biomarker computation. So it has a lot of downstream applications for cell detection. We want to solve this problem. In the current literature, you probably find many studies that show that they use or train a CNN model or convolutional, fully convolutional neural networks, FCN, to do this cell detection. And usually training this kind of CNN model will require a large amount of annotated image data. For example, you will require to label the position of the nuclei for all of them in these images. And you can image that this is very time consuming, or as we know, the data annotation in the medical domain is very expensive. So in this case, it's possible that you will lie to the cell detection in a data set that they don't have the label data. So how can we train a model or train a state-of-the-art CNN model or other deep learning model on a data set that they don't have the label images? So how can we train this kind of model for the automatic cell detection? And here we introduce a GAN-based model for the domain adaptation for the cell detection. And the idea is based on GANS, or generative adversarial networks. So this idea is very interesting. The GAN framework has two components. It has a generator and a discriminator. The generator will take the random noise as input, and then it will generate the fake images from the random noise. And on the other hand, you also have the real data, right? The real images, but you don't have the label. You can you feed this real image and fake image to the discriminator, and this discriminator will try to classify these two types of the samples. It will differentiate that, oh, this guy is a real or is a fake image. So you train this model by do a minimum and max scan in this kind of GANS framework. After the model training, and the discriminator will not be able to differentiate these two types of images, which means that the fake images are very similar to the real one. So when this is fake image you generate from the random noise or some other image from other domain. So you can use the fake image data for model training, although you don't have the label for the real data. So this is the idea for our method. So this is the architecture we propose. And suppose that we have a two data set. source and target. And for the target data set, we will lie to the automatic self-detection in this target data set. But as we mentioned, we don't have the labels in this data set. We don't know where are the cells in the training images. So we are not able to directly train the state-of-the-art deep learning architecture for this self-detection. But in the source image data set, so the image that which is a public data set, they have the annotation available. This is the H&E standing image column cancer. It has each nuclei annotation and labels in the image data. So can we train a model using these source images and then apply model to the task image data for self-detection? If we train a model directly using this source image data, probably the model will fail in these cases because these two images look very different. They're very, in terms of the color and intensity, right? So it will probably fail in these kind of cases. But based on GANS, we can transfer the images, transfer the source images to the target style image look like this. See, we use the kind of generator to transfer these source images to the target style image data. And this transform source image will look similar to the target image look like this. And of course, here we will need a discriminator to differentiate these two types of images. And we will train this model until this discriminator is not able to differentiate these two types of image data. Then you are done. You can train a model state-of-the-art deep architecture using these images, then apply the model to the real target image data. And because this source image data, we have the labels. And after the image transformation, the label should be the same because if we have a cells here and after do the image translation in the same position, you also have cells here. So the semantic information should be preserved. So then, which means the label should be the same, but the images change. So you can train the state-of-the-art architecture using these transform images and their labels, and then apply model to the target data set. So that's the idea for our method. And this is some mathematical details. Suppose that we use the GST to represent the generator for the source to target domain mapping, and the D is the discriminator. Here we would like to use this G, the generator, to transfer the source images to the target style image data. Then we use this discriminator to differentiate the transform source images and the real target image data. Then we will minimize this loss function to learn G, but simultaneously to maximize this loss to learn D. And after model training, you will get a very nice generator for the model image translation. And on the other hand, we also learn another pair of generator and discriminator for the target to source mapping integration as well using this function. This is the basic idea for the GAN loss. And in addition to ensure that the reconstruction of the images should be identical to the original images data, we apply a cycle consistent loss as well, which means that if we apply or invert transformation to the transform the source image data, the result should be the same as the original image source image. So the benefit for this cycle consistent is that it does not require image pairs. So you can image that. This is very important for the self-detection in cross modality in microscopic image data. For example, if you have covered H&E stand images, which is from the breast cancer tissue, and you have another data set, which is IHC stand, which is from the pancreatic neuroendocrine tumor tissue. So this cover data, they are not paired. They are even done from the same patient. We don't have this kind of image pair for model training, and this cycle consistent loss can help us to avoid this problem for the self-detection. And finally, we'll combine all the loss in this kind of single function. And here we just to do use a L1 penalty for the reconstruction for the cycle consistent loss. So this is the GAN loss, we combine the GAN the loss in the previous slide and this cycle loss as well to train the GAN. And this is the GAN framework. It will transfer the image between source and target domain, but we still need an architecture to do the cell detection. Remember that? Because the GAN framework, they only perform the image translation. They don't do the cell detection. So here we use one of our previous work, which is based on the fully convolutional architecture, and it has a encoder and decoder components. And this is the deep regression model. The decoder will learn the features from the input image data, and the decoder will map the feature map into the space of the input images, such that the output of the prediction map will have the same size of the input images. And in this prediction map, the local maxima will present the position of the cells. So this is kind of the basic idea for the this deep regression model. And for the loss function, we introduce the weighted mean square error loss function, because probably you can, from the previous slide or maybe from these slides, you see that the image background has much more pixels than the nuclear region. So here we would like to emphasize or highlight the contribution of the nuclear or the cell region during model training. Otherwise, it's likely that the model will just provide a trivial solution by predict everything as the background pixel. So we don't want this kind of solution. We just use a weighted mean square error loss to train this regression model. And finally, we combine all the loss into a single function. Then we train this function for the cell detection. So after we train this model, we expect that the model should be able to apply to the real target image data, because this model is learned with the transform source images, which are target style image data. They are very similar to the real target images. So this is the kind of the basic idea. So in the second step, we introduce an additional stage for the modified tuning based on the procedural labels. And remember that in the first stage, we only use the transform source images for model training. We do not take advantage of the real target image data because we don't have the label for the real target images. But now we have the model. We have the model which is trained with the transform source images. We can apply this model to real target training images to generate the procedural labels. And we hypothesize that using this kind of the procedural label, we can further improve the model. And because it will use both gold standard and annotated source images and high quality real target image data for model training. And we apply this method to different three types of microscope image data. And the first one is the age standard colon cancer and image data, and the second one is the KI67 IHC standard pancreatic neuroendocrine tumor, and the last one is cervical cancer face contrast microscope image data. So we do the domain updation on these three data sets. And before we show the results, I would like to discuss the kind of metric we use for the model evaluation. And this is different from the image classification, which is very straightforward to define the true positive or false negative, because you just to check whether the predict label is correct or not. For the object detection is a little bit different, and as we define the precision recall and inference in this way. So for each annotation point in the images, we define the gold standard region, which has the center in this annotated point. And for any prediction inside this gold standard region, we will say that this is a true positive. Otherwise, it will be false negative. But it's possible that we will have a multiple prediction in the same region. So here we will use some kind of the Hungarian algorithm to do the optimization. Here we build a bipartite graph, and with the nodes will be the prediction and the gold standards annotation, and the edges between the nodes will be the distance between the prediction and the gold standard. And we solve this optimization problem by using this kind of the Hungarian algorithm by minimizing the total loss. And after we do this kind of the nodes assignment, we can define that for the predictions, if they have the associated gold standard annotations, they will be true positive. Otherwise, it will be false negative. For those gold standard annotation, if they don't have the associated predictions, they will be false negative. So based on this definition, we can calculate the precision we call an F1 score. Okay, so this is some kind of results. And this is original, the first column is the original image, and the second one is the gold standard annotation. And this annotation is manually by the pathologist. And the last two columns is the automatic cell detection results. And for example, for this IHC to H&E standing image data, for these cases, the third column, we transform the IHC standard images to the H&E standard images for model training. Remember that for these two columns, we don't use any real target annotation for model training, but you can see here, the results are very promising. And here, this is another result for the data set of IHC standard data. And the last one is apply the model to the phase contrast image data. And for this image data, you can see that compared with the ground truth, we almost correctly pick every single cell in this using our method. And we also apply our model in a semi-supervised documentation situation as well. In this case, we assume that we have a limited target annotation available in the target domain. Then we combine transform the source image data and the real target image data for model training. And this is the F1 score for the cell detection. From the figure, we see that using only very limited real target annotation, we can get a very kind of competitive performance with those models that using all the real target annotations. For example, for this one, you're only 40% real target annotation, we can get the very equivalent performance for those fully supervised learning model. From these figures, we probably can reduce the human effort significantly for the data annotation in the target domain. So this is kind of our previous project for the automated cell detection. And we also recently extend this work to the cross-site consistent documentation for the cell detection as well. But we apply this model to the data set which are from different labs, even though this kind of image come from the same tissue, as shown in these two images. These two images are, both of them are from the pancreatic tissue, but they look very different because of the inconsistent imaging process or using different device. So if you train a model for cell detection or identification using the images from lab 1, probably it will fail in the lab 2 because the intensity look very different. The distribution are totally different in these cases. So here we further improve the previous method by introduce a cross-site consistent constraint, which is shown here. So remember that in our previous study, we don't have the label for the target image data, and we train a target model by using the transform the source images, such as this one. We transfer these source images to the target style image data, and then use this image data to train the target model. But we add additional constraint which is called cross-site consistent, which means that although we don't have the label for the target images, but the prediction for the target images before and after transformation should be the same or should be consistent, because we can use the GAN model to transform the target images to the source style image data, and in the source domain, we have the data annotation for this one. We can train the model to do the prediction for this transform the target images, and the prediction look like this. And also on the other hand, we can train a task model in the target domain by using the transform source images, and this is the RT. So if we apply this target model to the real target images, and we also apply the source domain model to the transformed target images, the prediction should be similar or should be the same, because during image transformation, the content should be preserved. Only the style will change, the color or in texture will change, but the content will be, or semantic information should be preserved. So that's the motivation for this cross-site consistent loss. We have some kind of preliminary results for this task. Here we apply this using our method to do this kind of cell detection and classification, and compare this kind of the model that don't use this cross-site consistent loss. We can have some kind of about 5 point improvement in terms of F1 score. So for this kind of the application, we again use the GAN-based model, but we introduce an additional constraint for the image transformation. Okay, so this is the GAN-based image analysis in our research. And the next project I would like to discuss is Ki-67 labeling index assessment. And Ki-67 labeling index, or the Ki-67 score, is one kind of the biomarker which can be used to determine the grade of the pancreatic neuroendocrine tumors. And this score is defined as the number of immunopositive tumor cells over the number of total tumor cells. So in order to compute this score, we will need to differentiate different types of cells in the images. We need to classify the immunopositive tumor cell, immunonegative tumor cell, and non-tumor cells as well. So in order to compute this score, we present the end-to-end architecture, which is based on the fully convolutional architecture. In this architecture, we have 2 branches. The first branch is the cell identification. In addition to the cell localization or nuclear localization, we also need to classify which type of these cells is the positive tumor or negative tumor or is non-tumor. And in addition to this branch, we also have a second branch, which is called region of interest segmentation or tumor region segmentation. The motivation for this design is that when the pathologist determine the class of the cell or the nuclei, they not only look into the cells themselves, but also inspect the surrounding regions, such as the spatial distribution of the cells. It's possible that tumor cells are close to other tumor cells and non-tumor cells are close to other non-tumor cells. So this kind of the context information is very helpful for the cell classification. We hypothesize that if we can segment this kind of tumor region, and they can provide this kind of the an additional information for the cell identification, we believe that they will be helpful. So this architecture is kind of the end-to-end architecture. So we will simultaneously learn this architecture by learning these 2 tasks, the cell identification and also the region segmentation. And we will minimize the 2 losses. And the first one is the cell identification, which is just the deep regression loss. We use the one as we discussed a few minutes ago. And for this kind of the region segmentation, here we use kind of the mask guide cross entropy loss. Because for our region segmentation, we do not require pixel-wise notation because that is very expensive. We only require the random region annotation in the tumor region and non-tumor region and a subset of the region for notation. So this count is only adds only a small overhead for data labeling. But with this very small overhead, we can get a better result for the cell identification. will minimize these two losses simultaneously. And after the model training, we can apply the model to the model testing. And given the input image data, it will directly output the positions of the nuclei or cells, and also output the labels, the class of the cells as well. And during the testing stage, the segmentation of the tumor region is not necessary. So usually we will discard the second branch during model testing. Here we show some kind of the results using our architecture, single stages KICD scoring, and different color means different type of the nuclei. And we also compare our method to some other deep learning architectures, including those very popular deep neural networks, such as the UNet architecture, and as well as the fully convolutional neural networks. We also compare our method to the recent state-of-the-art deep learning-based method based on deep regression modeling. And based on the table, we see that our method can provide the best performance in these tasks. Okay, so this is another project which is using this kind of the multitask learning based on deep neural networks for the KICD7 label index assessment. And the final project I would like to discuss today is liver lesion detection in the paired image data. And for example, this is one kind of the images here, the paired images. And in this paired image, we have a kind of the lesion in this here, and we draw the circle here. We would like to develop a deep learning algorithm to automatically locate this lesion in the paired images. And the original data set are 3D. Here I just show one slide here. This project is ongoing, and we are still working on that. And this is the current architecture we are using right now. And this architecture is based on 3D UNet. And here we replace the traditional convolutional layers with the residual learning block, because residual learning block is more effective, and it has been used in many other studies and show improved results. Here we also introduce two additional context aggregation path here, because the neuron in different layers will have a different size receptive field. It can only see a local region in the input images data, but we can have a different size of a lesion, right? So here we hypothesize that if we can do the information fusion of multi-level feature maps, we expect that we can handle this kind of scale variation of lesion in the input image data. So here we conduct the data fusion after the last residual learning block here. And then finally, we have some kind of the traditional convolutional layer for the lesion detection. In order to train this model, we asked the radiologist to provide the label data. And the label images is just binary labeling. And if the pixel is inside the lesion, it will be one, it will be other will be background, be 0. So based on this annotation, we can use a standard or traditional cross entropy loss to train our model based on the back propagation algorithm. And the Y here, just the label, and is the one-hot vector to indicate the pixel is inside or outside the lesion. We have some preliminary results for this project. For example, for this patient, it has the lesion here, and this is the ground truth. And then the model can correctly detect the lesion. And also we have another one, and this patient have a much larger size lesion. But as you mentioned, we can handle this kind of different size of lesion in the images. And the last patient is that this patient have multiple lesions in the liver. So here we showing that we can identify 4 of them. We still miss one, but I guess later on we will further improve this model, and hopefully we can get a better results based on this architecture. And one last thing that I want to mention, currently this data set, we are still collecting the image data. We only have less than 50 patients for this project yet. So we will continue collecting data and then improve our model. And once we believe that if we have more and more data, I think that the model will have a better performance. So I think that's all for our discussion for today. So for now, I will happy to take questions. Thank you.
All right, let's thank the speaker either by sound or reaction. Great job, Fuyong.
Thanks.
Yeah, so any questions from the audience? Again, you know, feel free to use the chat box, or if you feel brave enough to unmute yourself and ask a question, please go ahead. So I'll start with one. So with these deep learning algorithms you're fitting, you know, they're pretty, you know, data, for them to work well, you need sort of, like you kind of alluded to, a lot of data.
Mm-hmm.
I'm wondering, like, one thing people have sort of shown is that sort of synthetically augmenting your data set through data augmentation can actually be very helpful for performance. Are you doing sort of data augmentation steps in any of the projects you've talked about so far?
Yes, actually, for the project we are using GANS. Actually, is one type of data augmentation technique. You can generate the fake target data set and to train a model. And this is, I think, this is for the generative model or GANS, especially for the GANS model. I think this is a kind of a very popular data augmentation technique we are using right now. And in addition to this kind of GAN-based data generation, we also have some many other such as the color augmentation and also this kind of the geometric transformation, the traditional data augmentation technique you can use. But I think now the generative model, especially for the GANs, I think is kind of very popular.
So another question I had for you too, again, other people can ask questions, but since no one's asking, I'll ask. You know, could you talk a little bit sort of about sort of the computation sort of required to do this? And, you know, and my sense is, you know, data sets will get bigger. And I'm just wondering if you could say a little bit about of whether you think things like cloud computing are going to sort of become more popular in this field going forward.
Sure. Yeah, I think this is a great question. And for the kind of deep learning model training, usually we'll need very powerful GPU for model training. And as you mentioned, maybe use the cloud in Google Cloud or Amazon, AWS cloud computing. And for this kind of I can give an example for when we train the GAN model. I think at that time we set the batch size as 1 because we don't have that large memory to fit the, let's say, more than 1 pair image in the memory, GPU memory for model training. But of course, if you have the large scale computing resources such as you can use Google Cloud, I think they will be very helpful. And I think in our campus, we have the Eureka computing system we can use for this kind of things. But if of course, the Google Cloud is not cheap. I know the price is really high, but for the personal use, I will suggest that just purchase your own machine for deep learning boards. And I guess right now for this kind of the GPU prices, $1,500, we'll get a very nice GPU. We'll have a 24 gigabyte memory, and with 24 gigabyte, I think you can handle most of the tasks you are working in your own research. Otherwise, if you want to be Google or Microsoft or Facebook, this kind of big company, you want to build this kind of the really big, huge model using image in the data set, you will need cloud. But maybe in your own research, we don't have this kind of large image data set. And probably the 24 gigabyte memory, I think, is suitable for most of the applications.
All right, thank you. Here's a question from the chat box. Have you used these machine learning algorithms on any multispectral data from the Vectra or Polaris or MIBI machine?
Yes, I think this is kind of nice question. Actually, no. I don't apply this kind of algorithm to this multi-spectral data, but I think from the technical viewpoint, the concept should be the same. Just the data will be different, such as the modeling, the deep architecture building, how many layers you need, and then what's the loss function. All these kind of technical concept will be the same. Just the data, or you probably need to tweak a little bit of your architecture to fit your data set. So, but I don't think this will be not a difficult problem for this kind of changing.
And you see another question. So I think these are you had pictures of slides with true cells and predicted cells. It says in the clear scoring process, there were often closely juxtaposed nuclei where only one of the nuclei were scored.
Okay, so
you have these. I think it goes back to the slides you had where you had pictures of the slides and the true cells and the predicted cells. So it looked like. I mean, so could you maybe I'll sort of paraphrase this question. Could you sort of comment on the ability of deep learning algorithms to sort of correctly classify sort of nearby cells that are like really close to each other,
okay,
rather than only picking one?
Yeah. Okay, I think this is kind of very good question, and actually this is a challenging question in the computer vision and image analysis. So if this kind of two cells are too close, and then probably you only recognize one. Yes, we do have this kind of technical to handle this. But usually this will based on the segmentation, because by segmentation it will be much more challenging than the detection. And for segmentation, you will need to require to delineate the cell boundaries, and if the two cells are close to each other, so you have some kind of the algorithm to you can successfully separate this kind of the close or touching or overlapping cells. We have this. I think in the deep learning community domain, the people publish a lot of paper on this, and actually one of them we are using right now is called horizontal and vertical distance map. So in that we calculated the horizontal edge map and also the vertical edge map as well, and use this information to separate the touching or even partially overlapped cells or nuclei. I think you can do this, but right now, as you mentioned, this question is not solved yet. So it's very challenging how to handle this kind of the touching or scale variation in the pathology images data set. This is still an unsolved problem.
All right, thank you. Other questions?
Okay, I think seems
okay. Again, thank you again, Fuyong, for the great presentation. I forgot to mention one thing. I see Toby Cornish on here. So actually one of Fuyong's accomplishments that I should have mentioned at the beginning was he and Toby, they've actually been part of this national consortium called the Informatics Technology for Cancer Research consortium, which is basically looking at developing machine learning tools across a variety of cancer domains. And they've actually had an R21 funded the last couple of years to work on this. And I think some of what you've talked about today was sort of based on proposals in that. So
based on the R21 project.
Yeah. So we keep looking forward to seeing more good things from you and Toby and collaborators coming out. So, okay. All right, everyone, thank you. Thanks again to Fuyong. And everyone have a great rest of your day. Okay. Thank you, everybody.
Thanks. Bye.